Circular 15. Tonight we are discussing a scientific topic that is going to break the dogma of uniparental mitochondrial inheritance. Tonight we have with us a researcher specialized in this genetic file, Roberto Grosso. Give it up for Mr. Grosso. Hello, how are you?
Hi, I'm very excited to be here, thank you.
Okay, alright. So, how did this project begin?
Okay, so everything started when we met a boy in Cincinnati who was suspected to have a mitochondrial disorder, and then none of his relatives showed this mitochondrial disorder. So then we decided to sequence his DNA, his mitochondrial DNA, along with his sisters, because he has two sisters. And later we compared it to the reference. We found out both homoplasmic and heteroplasmic variants in their genome. So this means that his mitochondria and his sisters were not all the same. So we think all of this is inherited from their mother. After evaluating his mother, we discovered that she had mitochondrial DNA both from his mother, which was the kid's grandmother, and from his father, which was the kid's grandfather. With these investigations, it could be possible to demonstrate that biparental mitochondrial DNA transmission exists.
Wow, that's so interesting. Then why was this case the inflection point? Why it isn't the normal situation?
Because the traditional knowledge considers that paternal mitochondria are all eliminated in the embryo. However, we think in this case, this mechanism of elimination has failed, and that's what we are trying to know why. Nevertheless, even when some paternal mitochondria are not eliminated, they usually end up dissolving because they are in a minimal proportion compared to the maternal ones.
So, if they always end up disappearing, how did they find them?
So our hypothesis is that the mutation not only avoids paternal mitochondria DNA elimination, but also needs to motivate its replication. We already have evidence that this replication exists, but we need to know and explain how the maternal mitochondria DNA level remains untouched even when this paternal mitochondria DNA replicates.
Okay, thank you so much, Mr. Wilson.
Thank you.
Goodbye.
Goodbye.
This huge discovery has important consequences in the field of clinics and human evolution. Tonight we have with us also a Roberto colleague called Blanca Garcia. Welcome, Ms. Garcia. And what are you going to explain us, Ms. Garcia?
So, in clinics, if we found nuclear genes that regulate uniparental inheritance, we could induce paternal mitochondrial transmission in order to have healthy offspring without a female mitochondrial donor, or without using the three parents technique. The discovery of these genes would also help to develop new treatments. And this discovery would also have some important consequences in the human evolution history, because mitochondrial DNA have been used as a reference for this. If we take into account these new studies, our mitochondrial age could be 4 times older than we have considered before.
Thank you so much, Blanca, and goodbye. And goodbye, everybody, and thanks for watching Circular 15.